Good afternoon, everyone. Welcome to my talk on SCB 2021 annual conference. My name is Kalyanikrishna. I'm a PhD student at AG Schubert Epigenetics of Plants, FU Berlin, Germany. First of all, I would like to thank the organizers of SCB and InDepth session for giving me this opportunity. Today, I'm going to talk about my PhD topic, which deals with the study of a SWI chromatin remodeler complex subunit that interacts with PWO1 and CRWN1 to regulate nuclear morphology and chromatin organization. Once again, welcome all of you. We all know that the eukaryotic cell nucleus is a phenomenal organelle that is a site of storage, amplification, transcription, translation, and regulation of vital information that is needed for the proper functioning of the cell and the organism itself. In order to achieve that, the genome inside the nucleus should be arranged in such a way that it provides a landscape for dynamic and differential genome regulation. And this is controlled or mediated by several levels of checkpoints, starting from the linear DNA sequence to the actual 3D genome positioning within the nucleus. One of the key factors that plays a role in this is the subnuclear compartments, which includes the nucleolus, nuclear bodies, Kajal bodies, nuclear pore complex, and the nuclear periphery. Today, we are going to talk about the nuclear periphery, which includes the nuclear envelope and the nuclear lamina. Nuclear envelope is a double membrane structure, and it consists of an outer nuclear membrane, inner nuclear membrane, and the space in between. In a typical metasovan cell, there is a nuclear lamina, which is seen just beneath the inner nuclear membrane, which appears like a mesh-like structure, and it extends the nuclear plasm. So it is acting as a connecting bridge between these two. The nuclear lamina is composed of coiled structural proteins called lamins, hence the name lamina. It is well studied and well characterized in animal kingdom. Nuclear lamina, along with the periphery, is known as the gene silencing hub of a cell, as it harbors tethering sites for silenced chromatin or chromatin with heterochromatic features like H3K27 trimethylation mark. When we take this scenario into plant cell, there is no homologue for lamins present in plants. So it has been believed that lamina is absent in plants. However, throughout these years, extensive research has shown that indeed there is a similar network of fiber-like structure just beneath the inner nuclear membrane in plant cell nucleus, and it is encoded or it is formed by plant-specific proteins called crowded nuclear proteins. These are encoded by 4 genes, CRWN1, 2, 3, and 4. Today, I'm going to talk about CRWN1, which is localized to the nuclear periphery, like a ring-like structure. And the mutants of CRWNs doesn't have a definite phenotype when we look by eye. They look similar to the wild type. However, when we look at the nucleus, the nuclei are smaller and more roundish compared to the large elongated wild type nuclei. And then the questions were like whether crown act similar or function similar to that of lamins, whether they helps in preferential localization of chromatin to the nuclear periphery, they play a role in chromatin organization, and so on. Recent studies from 2017 to 2019 has shown that indeed crowns have a role in chromatin organization, and they harbor sites for tethering chromatin with heterochromatic features. And also, plant lamina-associated domains have been identified by using CRWN1 as a bait. So this points in the direction that plant cell nuclei also has a similar structure like animal cell nuclei. However, it has plant-specific proteins that plays a key role in chromatin organization and forming the plant lamina. One of the key factors that is regulating gene expression by putting heterochromatic marks are PRC2 complex or polycomb repressive complex 2. And previously in our lab, we have identified a protein called PWO1 that interacts with PRC2, which is shown to be interacting with CRWN1 as well. So PWO1 pulled down CRWN1 as one of its interactors, and then we confirmed this interaction by several protein-protein interaction studies. And then we looked at the nuclear morphology of PWO1, and it has been shown that PWO1 regulate the nuclear morphology. Like in PWO1 mutants, the nuclei size are reduced. Then we looked into the RNA-seq from PWO1 and CRWN1 2 double mutants, and we have seen that there is a significant overlap of the misregulated genes indicating that PWO1 and CRWN1 regulate similar set of targets. And in the PWO1 up and CRWN1 2 upregulated data set, half of the overlapping genes were also targets of H3K27 trimethylation. And these were, and by gene ontology, we could identify that these were mainly genes related to stress response or stress-induced genes. In order to further characterize the PWO1 CRWN1 interaction, we decided to look onto more interactors of these two. So when we overlap the PWO1 interactor list with the nuclear lamina enriched fraction, we could see more than half of the PWO1 interactors were overlapping with this lamina fraction, which included CRWN1 and CRWN4, and also several chromatin associated proteins. For example, SWI SNF complex subunits, uh, ARPs like actin-related proteins, CHR17, PDS5, etc. The SWI SNF complex being one of the largest chromatin remodel complex and a well-conserved complex across taxa spiked our interest, and we decided to look more into that. And one of the subunit from SWI/SNF complex that was found in this overlap was ATBOB60, also known as CHC1, SWP73B, 73B in yeast, or SMARCD2 in humans. It is part of the large complex and it is essential for its function. So SWI/SNF complexes are basically ATP dependent complexes. So it has ATPases, for example, Brahma and SID, and they are essential for plant development. For example, ATBOB60 knockdown has a line, it's a tDNA line, and it has shown that it is essential for the proper development of plant. Otherwise, the plant is severely affected with dwarf phenotype, late development, twisted leaves, and so on. And this can be complemented by introducing a 35S ATBOB60 YFP construct. The plants were rescued. Then we confirmed the ATBOB60 interaction with PWO1 and CRWN1. So indeed, ATBOB60 showed the interaction with PWO1 and CRWN1 via yeast-2 hybrid and FRET analysis. And then we checked if there is a genetic interaction by crossing ATBOB60 with PWO1 and CRWN1. So PWO1 single mutant and CRWN1 single mutant phenotypes are not so different from pol0. They are not severely affected. However, ATBOB60 mutant is severely affected, and the double mutants show further enhancement of this phenotype. So the genetic interaction between these interacting partners were also confirmed. Next, we looked at the nuclear morphology, which is the key phenotype in these mutants. And we could observe that ATBOB60 indeed showed a reduced nuclear area and increased circularity, just like CRWN1. And in order to support this, we also tested in order to see whether it is a peculiar phenotype shown by ATBOB60 or whether this is common for the SWI/SNF chromatin remodel complex. We tested other subunits of the complex, for example, Brahma 5 allele, SWI3C, CHR12. And as you can see, SWI3C and CHR12 doesn't have a phenotype that could be observable by eye, but still, all of these SWI/SNF subunits show similar nuclear morphology as ATBOB60 and CRWN1. Then we looked onto the RNA-seq of ATBOB60 and PWO1. Here we collaborated with Musa Benhamid for the RNA-seq and chip-seq experiments. And when we looked at the commonly misregulated genes, we could see that ATBOB60 and PWO1 overlap quite significantly. And in addition, 17 out of these 31 commonly upregulated genes in BOB60 and PWO1 are also targets of H3K27 trimethylation. Then we looked onto PWO1 and ATBOB60 targets from chip-seq. And it was interesting to note that more than 60% of the targets of PWO1 overlap with that of ATBOB60 targets. So these data suggest that 80 and PWO1 appears to function in the same pathway. As part of the STSM funded by the InDepth, I was given the opportunity to work with Celia Baruch in University of Zurich, where we focused on confocal and super resolution microscopy with immunofluorescence studies and also 3D image analysis with MRS. We focused on the mutants I was using for my thesis. And when we did the DNA dye staining with a 580 CP oxide dye, we could observe that there are that this dye is more sensitive than DAPI, and there were structures outside of the chromocenter uh, that are stained darkly, and however, they are smaller than chromocenters, which were termed as non-chromocenter chromatin or non-chromocenter heterochromatin here. And then we did 3D image analysis by segmenting the nuclei. We created a nuclear mask, and then we segmented the chromocenters and the non-chromocenter chromatin. And as you can see, the chromocenter organization, chromocenter distribution, and the chromatin organization in 80BAF60 is quite different from the other genotypes. And we could see a lot of non-chromocenter chromatin foci in 80BAF60 compared to the other genotypes. And the chromocenters are really conspicuous and big in other genotypes, but this is not the case in 80BAF60. So this indicates that 80BAF60 is somehow affecting the chromatin organization. Further qualitative and quantitative analysis on this data is going on. I also measured the relative heterochromatic fraction in this nuclei and found that in 80BAF60, there is a reduction in the relative heterochromatic fraction. So these results indicate or points in the direction that 80BAF60 is essential for proper chromatin organization inside the nucleus. So the model we propose here, we can see that the nuclear periphery or the nuclear lamina in plants composed of the crown one interacts with two major complexes within the nuclei. One is the polycomb repressive complex with the with PWO1 as its interactor, interacts with crown one. Crown one also interact with 80BAF60, which is part of the SWI/SNF complex. So, and these are indeed PWO1 and 80BAF60 are interacting with each other. So these three are interacting together and plays a role in chromatin organization and regulating nuclear morphology of Arabidopsis. In order to give a scenario, it could be like that. When there is no stress, the stress-induced genes are marked with heterochromatic features and localized to the periphery with the help of PWO1 and the SWI/SNF complex. And when there is a stress and when these needs to be activated, chromatin remodeler complexes can act and modify the chromatin accessibility and helps in activating these genes. This could be one scenario, or there could be other mechanisms that they can act independently also. And with that note, I would like to bring your attention to three studies that were recently published. One is from 2011 in PLOS Genetics, which states that SWI/SNF components are associated with lamins A or C, and also lamin A or lamin A and C regions binding chromatin were also found to have H3K27 trimethylation marks. Another study from 2013 from PLOS1 shows that the SWI/SNF ATPase BRG1 induce nuclear shape changes which are independent of cytoskeletal connection, and the expressions of lamins are also not changed. Another study, which is quite recent from 2020, shows that the NMCP1, which is equivalent to crown 1 in Arabidopsis in rice, interact with the SWI3C to regulate drought resistance in rice. So these studies also support a similar hypothesis that we have. With that note, I would like to thank my supervisor, Daniel Schubert, for all the help and guidance, all members of Schubert Lab, people who worked on the project before, and our collaborators, Celia Baruch and Musa Ben Habib, for all the help and guidance. Thank you so much. And thank you for all for listening.